thank you very much, James, for joining us. James, to be honest, doesn't need any introduction to anyone that works on machine learning for healthcare at the cutting edge. I really do not need to introduce James to any of you, but I'm going to do it just in case there is somebody coming more from the clinical side or maybe more from the technology side or biology side. So James is really a superstar. He is an associate professor in Stanford. And James and I met quite a few years ago in Stanford, and I'm a huge, huge fan of his research and his group's research. He received numerous awards, not only the easy ones, but also the very amazing ones like Sloan Fellowship. He has 2 Chan Zuckerberg Investigator Awards, numerous Best Paper Awards, faculty awards, has been profiled in popular media from New York Times to Wired. And my group is a very big fan of James' research, and every time he is publishing something at one of the big conferences or one of the Nature journals, we are reading it with great pleasure and learning a lot from it. So you are up for a big treat today. I hope you will enjoy it as much as I do. And if you are one of my students, please pay attention and learn from James. Thank you very much, James.
Great. Well, thank you so much, Mihaela. It's always really lovely to interact with your center, your group here, and really excited to meet everyone here. And also, I'm a huge fan. I was mentioned to Mihaela, I'm a huge fan of all the great work that you folks are doing. So congratulations, and looking forward to exploring a lot of different collaborations. Yeah, so I'm really excited to be here and to share some of the work that we're exploring. I think in a really exciting space, and I know that many people in the audience are also exploring the space around generative AI, large language models, and so on, and really thinking about how do we develop and apply these generative AI tools to solve important and impactful biomedical problems and applications, right? And then we're really going through this, I think, a really quite exciting transformation, right? Both in AI and also in applications of AI. Right, and a lot of that transformation is really driven by the rise of these very powerful technologies like large language models, right? And this is, just one snapshot of the landscape of these different generative AI large language models. And as you know well, there are new models that are coming out basically every week or every couple of weeks. So today I want to specifically think about how can we actually use these generative AI tools and develop these tools to really help to solve biomedical problems, right? In the end, I'll say a little bit more about what are some of the core challenges on the AI side behind these tools that we also want to address. But I want to really focus on some of the biomedical applications, given the interest of the people here in the audience. And in particular, when we think about biomedical applications, right, it's important to think about what are the problems for different stakeholders. Right, and usually there's sort of 3 main groups of stakeholders that we're often interacting with as AI researchers. Right, so there are biomedical researchers, right, who could be biologists or other people who develop these algorithms. There will be clinicians, and then also importantly, there are also patients. Right, and each of these 3 groups of folks have their own problems and requires different kinds of AI solutions. And today I want to share with you some of the areas we're exploring around how we can use generative AI to try to benefit each of these 3 groups, right? So that will be the 3 parts of the talk. So first, I'll say a bit about how we can try to use and develop new kinds of generative AI tools to help researchers. And in particular for researchers, I think one of the big promise of generative AI is to really help us to expand the search space in some sense, right? The creativity of researchers. So I'll give you a concrete example of that. And then next, we'll talk about, from the perspective of clinicians, right, how we can actually build generative AI tools, in particular chatbots, that can be an assistant, right, a personalized assistant for clinicians to help them to make diagnosis, to help them to interpret cases and images. And lastly, from the patient's perspective, I think one really exciting application of generative AI is to really make medical information, medical knowledge more accessible, right, easier to understand for more diverse patients. So I'll give you an example of that. And so really appreciate that there's quite a people from different backgrounds here in the audience. So what I'll do is I'll pause after each of these sections, right? And we can actually have questions after each of the sections. But if you have questions at any point, also feel free to either put them into the chat, or you can also welcome to interrupt me if you like. Okay, so diving in. So first we'll talk about how we can try to build these tools for researchers. And I want to give you like a very concrete example just to ground this. So I'll tell you about how we actually build a more general algorithm for help us to discover new types of molecules and to create new molecules. But just to ground this in a very concrete application, I want to show you how we use these kind of tools to develop new drugs especially for antibiotics. So people might be familiar with some of the early history of antibiotics, right? So the first one was penicillin from the discovery in the 20s. And what's really interesting is that in the first few decades after discovery of penicillin, there's actually a gold rush, right? So people were discovering a lot of new antibiotics, right, from different sources. And that's great. But critically, for the last 60 years, there are actually relatively few structurally novel antibiotics that people were able to discover, right? And that's becoming a huge public health crisis, right? Because if we don't have a lot of new novel antibiotics, then it's easier for the existing bacteria to have developed resistance, right, to the existing drugs that we have. So currently, over 1 million people die every year because they're exposed to bugs that have developed resistance to the existing drugs that we have. And that number is expected really to grow very quickly over the next few years. Right, and one way to see this is in this kind of timeline we show here in the bottom, right? So above the timeline shows when a new antibiotic is actually first introduced into the market, right? And the bottom part of the timeline shows you when do we actually start to observe resistance, right, in some bacteria against those antibiotics. And you can see that in most cases, there's only a lag of a few years, right, between introduction and resistance, which really highlights the fact that we have this tremendous arms race, right, between drug developers and pathogens. And right now, we're not doing very well in that arms race because we're not discovering a lot of new drugs. And as a consequence, right, you have these very dangerous pathogens like the one shown here called Acinetobacter baumannii, that's considered by the experts to be the perfect predator because they have developed resistance to most of the current drugs that we have. So what I want to discuss today is how we actually develop sort of using generative AI tools to help us to create new drugs. So it's a very general technique that can be used for different kinds of drugs for different diseases, but I just want to go back to this concrete application of how we actually do this for antibiotics. And this work is led by Kyle Swanson, who's actually spent some time in Cambridge, is now a PhD student at Stanford, and is really tremendous PhD student that I'm fortunate to work with. Before I tell you what the algorithm is, let me tell you what is really the key technical problem that we need to solve. Right. So it turns out that it's actually not that difficult to have generative AI algorithms, right? Maybe diffusion models, other models that can hallucinate or come up with new molecules. Right. So that's actually a reasonable problem to solve. But the big problem is that the big challenge is that even though we can generate these new, we can have the model propose new molecules, but it's actually very difficult for chemists to actually synthesize these new molecules, right? We don't know how to make them. And if we don't know how to make them efficiently, then it's actually very hard to experimentally test these, and it's very hard to actually make these into new drugs. Right, so that's currently a big problem with generative AI in the drug discovery space, right? It's easy for these models to hallucinate new molecules, but it's actually very hard for us to physically synthesize these new molecules. So then what we wanted to do is to come up with a different approach where instead of having just the AI model hallucinate new molecules, but we want the AI model to actually generate for us a recipe for how to make each new molecule. Right? So it's going to give us the target molecule, like a novel molecule, but also the step-by-step instructions for how to explicitly synthesize those molecules in the lab. Right? And then we're going to basically essentially use relatively automated ways to synthesize those new molecules so we can experimentally test them and validate these. So let me give you an example of what it means to have a recipe for new molecules. right? So typically for these new molecules, we're starting with some building blocks. So you think of these as being fragments of small molecules that are widely available, right? You can order these fragments from different companies. And there are also some well-known chemical reactions, right? So you can think of each reaction as basically being like a template, right? For each reaction, you can put in maybe a subset of molecules, subset of building blocks or fragments. And by applying those reaction, right, then maybe you get more complex or larger molecules as output. So this is really not that different from recipes for cooking, right? You start with the raw ingredients, and you can apply different steps of the reactions to put these raw ingredients together, and you can get more and more complex outputs. But it's a very large search space, right? So the number of fragments that we have here that are easy to create and easy to that are widely available is on the orders of maybe over 100,000, right? And these reactions are on the orders of hundreds. And you can do this in different combinations. You can apply multiple reactions to multiple fragments. So you quickly have an extremely large, astronomically large search space of potential molecules. So the idea here is that the molecules that we have actually ever seen on the planet or ever have studied is only a very small fraction of all these potential molecules that we can potentially synthesize using these recipes. And the idea is then we're going to teach the AI algorithm how to generate new recipes, right, for these creating new molecules that we think are likely to be good drug candidates. So that's the high-level strategy. So then let me explain this in a little bit more detail in terms of the actual algorithm, right? So the first version of the algorithm that we have is inspired by these advances in game playing AI systems, right? So many of you are familiar with these AlphaGo kind of systems, right? Where they basically do, they have a very large game tree, and then they will have these kind of Monte Carlo search algorithms to go over the game tree to explore what are promising moves. We're going to try to extend that kind of approaches to the space of molecules, right? So in the space of molecules, we can also think of having like a very large tree, right? So here's an example of the tree where maybe at the level 1, I have the individual, what I'm calling these fragments, right, the building blocks. And as I go down the tree to level 2, then I can put together different combinations of building blocks, right? And then level 3, then I have the more complex molecules, right? So as I go deeper and deeper into this tree, I'm starting to put together different fragments, right? I apply certain reactions, and then I get more complex molecules. So basically, each path, if I go from the root to the leaf here, corresponds actually to an explicit recipe that I've shown you before, right? Like this path here that's highlighted in blue would say that, okay, let's take building block 1, let's add building block 2, apply reaction 27, and we'll get a more complex molecules output, and I can keep on going, right? So this is, I think, a very explicit but useful representation of the space of molecules, but also incorporates information on how we can actually synthesize these molecules, because each path now corresponds to actually one of these chemical synthesis recipes. So the issue here is that we want to explore this large molecular space, but it's just an exponentially large space. It's a very large tree. It's even much larger than the tree from playing Go or from chess. So how do we search this tree efficiently? That's where we need to have these AI to help us to guide this generation process. So one way that we try to guide the AI exploration is basically have like a reward model, right? So the reward model here, we call property predictor, is basically going to be a graph neural network that we trained to look at each of these molecules, right? It could be a molecule at any point on this tree. And then the reward model is going to try to give us like a prediction of how likely this molecule to be a good drug candidate. So the way that we train this reward model is basically through by doing first a drug screen, an experimental drug screen, where we experimentally test about 10,000 molecules, right, against different bacteria. And then we use that data to train this graph neural network to help us to basically make these reward predictions. And let's say when at one of the leaf nodes, when the reward model makes a prediction that says that this is likely to be a good drug candidate, that information then can be back propagated all the way up the chain, right, to update the individual fragments, right? So if the leaf is likely to be a good drug candidate, that also means that each of the fragments will get like a higher reward because they're more likely to be promising fragments. So that's how that encourages the general search model to explore these more promising fragments in a more efficient way. Of course, one challenge and one interesting challenge with drug discovery is that there's not just one objective, right? So certainly one important objective is this efficacy, right? How likely is this molecule to be a good drug candidate? But there are also other objectives, like there's structural novelty, right? We want the drugs we discover to be different from any of the existing drugs that people have seen before. We also want them to be safe in humans, right? And importantly, as we said, we want them to be very easy to synthesize at a high throughput. Right, so we basically can encode each of these different objectives as one kind of reward model, right? Similar to here, we have one reward model for efficacy, we have other reward models for safety and novelty, right? And this can all be combined into a single score. That's basically a score for the node. That's what we show here as the score. And so this was our initial setup, right? And we've since actually improved this model using reinforcement learning, right? Where the difference between reinforcement learning and the multicolor tree search is that the multicolor tree search does not explicitly leverage information across the different fragments, right? It's treating each of these fragments and building blocks as basically independent components, whereas in the RL setting, we can actually have an explicit value function that's trained across all of these different fragments. Right? So it's basically sharing information more explicitly across the different building blocks, different molecules. But the high-level idea is the same as we have here, right? Basically, we try to compute a score for each of these molecules, and the score will incorporate these different multiple objectives, right? And then we use those score to guide the exploration and the search of this very large molecule space. Yeah, so this hopefully give you an idea of our search strategy and how we set up this problem, right? So this is all done computationally, right? So the training data was come from some initial experimental drug screens that we had to train these reward models. And after that, everything else is done computationally, in silico here at Stanford, where we actually just have the model to generate in silico these new molecules. Now, of course, the fun part now is now we actually get to make these molecules and see do they actually really work. So to do this, we work with actually some fantastic collaborators in a company, biotech company called Enamine. So Enamine is actually, they specialize in making custom molecules, right? So the interesting part here is that they're actually based in Kiev in Ukraine. And as you can imagine, over the past 2 years, where they have a lot of other things on their mind beyond making molecules for us, right? But they were actually extremely good collaborators. So what we did is that we actually sent 70 new molecules that are generated by the AI algorithm. For each of these new molecules, right, the AI algorithm also generates an explicit recipe, right? So the recipe is basically just the path, right? It's like the step-by-step instruction of which building blocks to take and also which reactions to use, right? So we sent 70 of these recipes to Enamine. And because of the war in the background, they were not really able to optimize any of these recipes, right? So each recipe has to be extremely robust and very simple to follow, that they're able to basically just follow the step-by-step instructions from the AI model without any modification. And I think it's actually really quite remarkable and very nice that they were actually able to successfully synthesize 58 of these new molecules at a high throughput, right? Which means that the hit rate of these AI-generated recipes is over 80%. So that's already very good. And then it took us a while to actually get these new molecules, 58 new molecules, shipped back from Ukraine. But we got those back, and our experimental collaborators, John Stokes, is able to basically experimentally test how good are these molecules at actually killing these dangerous bacteria. And to do this, we focused on basically the most challenging bacteria, most challenging pathogens. So the World Health Organization has this classification of what's called escape pathogens, which are the pathogens. There are 6 escape pathogens. These are the 6 pathogens that have shown resistance to most of the existing drugs, right? So these are the most dangerous pathogens. And we had available 4 out of the 6 escape pathogens that we can experimentally test. So then what we did is that we actually tested each of the 58 AI-generated molecules against these 4 escape pathogens. And we found that there are actually 6 molecules that were effective against all 4 escape pathogens. Right, so these are the molecules that are shown here. Right, so each of these new molecules is is a new molecule that has never actually been seen before in nature. Right, so these are completely synthetic molecules that's proposed by the AI model, but they actually ends up being quite effective in killing these pathogens. It's not actually just not enough just to be effective in killing the pathogens, however, we also want them to be safe in humans, right? So we also have done further experimental validations to show that first that these molecules, they're very effective at a very low dosage. So this means that if the dosage required is low, this means it's more likely to be safe and less likely to develop resistance. We have also done further animal model studies, where we basically give these molecules to a mouse model and follow the mouse over time, and there's no sign of toxicity in the mouse model. So that's also a promising signal. And furthermore, we also track these animals over time, and there's no sign of or very low rates of pathogens developing resistance to these new molecules. Right. So I think this is still, definitely still needs a lot more experimental validation and then clinical trial studies. But I think this is already showing some quite promising signal, right, of these new AI-generated molecules that are shown to be effective in killing these pathogens, but also likely, it seems promising to have low toxicity in animals and humans. So this is the first example I want to mention, and to give you a sense of how we can try to use these generative AI tools to help to expand. It's really the search space, right, of chemists, of drug developers, to really explore new molecules that people haven't seen before in nature, right? And have this search space, and because the search space here is so large, that's where some of these techniques like reinforcement learning and reward modeling ends up being very useful to guide to the efficient search space, right? And I think the key insight here is that it's not just enough to find new molecules, but we also want to be able to find the pathway to synthesize those new molecules at the same time. And this is where these kind of tree-based modeling approach here we found to be quite effective. Okay, so then I'll move on to the second part, right? So we talk about researchers. So we do a lot of work also with clinicians, right? And one of the there's actually a question about the first parts. Divyash, do you want to ask your question, or do you want me to read it out?
James, maybe you read it out.
Okay. Okay, so the question I think I see is that, okay, the question is that to actually generate drug, different chemical reactions may require different environments, right? So how is the generative AI model able to account for these different chemical reactions and different needs of these reactions? So that's a very good question, right? Because we're looking for very robust pathways, synthesis pathways, we essentially try to build in a lot of domain knowledge into this AI algorithm. So the AI algorithm has a preference for using reactions that work reliably well at room temperature, right? And also at reactions that work well with different building blocks, right? So for example, if there are building blocks that are less likely to be effective with a particular reaction, then the algorithm actually knows that and knows not to select for those building blocks in going down this tree search, right? So it's a good question. So there's actually, we try to put in as much domain knowledge about the efficacy and the robustness of different reactions explicitly into this tree, into the AI model, so that it ends up picking these very robust reactions. And I think that's actually one of the reasons why the hit rate is very high when we sent these recipes to Enamine, right? Because they were not able to really optimize the different reactions, but they were able to still synthesize these at a quite high rate. So there's a second question from Melanie, also a very good question is, are we able to basically predict expiration dates for the new molecules, right? How long it might take for bacteria to develop resistance to new molecules? So that's a great question. So we're not able to predict the expiration date very well, because we don't really have great data, because these are really new classes of molecules, right? So we don't really have a lot of data to see how hard is it to actually develop resistance. The experiments that we've seen so far is basically tracking them on the orders of multiple weeks, right? Maybe over a month, and we haven't seen resistance yet. So maybe I'll say two things about this. First is that I think the fact that these molecules are structurally very different from the existing drugs makes it harder for pathogens to develop resistance. And the second thing is that we do expect at some point that the pathogens will develop resistance. However, this with the AI generation, the ability for us to develop these new molecules now is actually much faster. This whole pipeline, right, for developing the new, for the AI to train and to synthesize new molecules, to experimentally validating them, took about 2 months, right? Which is actually potentially much faster. So even if the drugs, when the bacteria develop resistance, we're potentially still able to come up with a lot of new molecules. Okay. Yeah, so Yu Dan has the next question, which is the AI-generated recipes are potentially based on our knowledge of existing antibiotics, right? So how does basically the algorithm make new molecules, or how do we potentially screen them for antibiotics? Yeah, so the reward model, to train the reward model with this property predictor, we have to use some existing experimental screening data that our collaborators had produced, right? So, and then once we have that reward model, basically, we're asking the generation model, right? To search through new parts of the search space, new part of the molecule space that we haven't seen before, right? So that does involve some amount of extrapolation from the training data to new molecules, right? So we're explicitly asking the model to go out of domain to search for these new models. And this is where actually some of these ideas around trying to quantify the uncertainty of these new molecules ends up being quite important. Okay, yeah, great questions. So then I'll move on to the second part around helping try to build assistance for clinicians. And I'll also give you a concrete vignette to illustrate this, which is based on some of our work collaboration with pathologists. Right, so pathologists are basically the clinicians who have to look at these images, right, from patients and try to diagnose what is the disease that a patient have, right? So it's often considered to be like the ground truth of medicine, right? Because they're the ones that have to make diagnosis, and the diagnosis then would inform what drugs, what treatments given to the patient. And it can be very challenging, right? Because they're looking at these high resolution microscopy images from different body parts, different patients, and there are all these heterogeneity, right, in different diseases. And oftentimes the pathologists actually frequently encounter images that they're just not familiar with. Right? So this is where we want to build an AI model to help them to interpret these images, and also that they can have conversations with. So something really interesting happens, right, when the pathologists encounter images that they're not familiar with, is that they actually talk about it on Twitter, right? So here's a few screenshots I took of these pathologists posting images, right, on social media like Twitter. And then you actually see a dialogue, right, where they invite their colleagues from around the world, the different pathologists to discuss and help them to interpret what's going on in each of these images. It might be surprising to some of you that your doctors and clinicians are actually posting these images online, but these images are de-identified, right? And it's actually for example, in the US Academy of Pathologists recommend that their members would use social media as a way to share knowledge with each other. And for our AI perspective, I think this is actually a really interesting and underexplored kinds of data, right? Because here you have both the high quality images and also dialogues, right? Multi-turn dialogues by experts interpreting these images, which is exactly the kinds of demonstration that we need in order to align AI models, right, to help to be useful and to interpret these images for pathologists. So then the first thing that we did was to basically curate this data set together as a resource. So the way that we did this is basically we go through all of Twitter, right, from 2006 until recently, and we use a specific set of hashtags that we know these pathologists use. These are actually hashtags that are recommended by the Academy of Pathologists for their members to use. So each hashtag will correspond to a different sub-area of pathology. And within that sub-area, then, like surgical pathology or renal pathology or cardiac pathology, there's like an active sub-community of people using these hashtags to discuss images. So we use those hashtags to get the initial pool of images, right? That's over millions of different tweets. And then a lot of those can be very noisy. So we actually implemented a lot of different filters to remove low quality images and also low quality text. So for example, we have another image classifier that we built to basically remove images if there's not enough high resolution or if it's too noisy or not relevant. We also have a text classifier, another machine learning model that we built to remove low quality text. So after all these filterings, then we end up with still a couple 100,000 of high quality Twitter threads, right? Where each Twitter thread will have one or more of these high quality images, along with the corresponding discussions by multiple experts around each image. So that's the data that we've curated. And this is basically just breaking down by the different hashtags, different sub areas, like how much data we're getting from each of the sub area, right? So for example, in some areas like dermatology or let's say breast pathology, right, we're getting quite a lot of information, right? But across many of these sub areas, we're getting quite a bit of data, right, on the orders of thousands of images and corresponding text descriptions that we can then use to help to align these models. Okay, and our goal here is to basically train a vision language model, right, that can help the pathologist to interpret these images. And and the key with these vision language models is that to really you have to try to learn how to align the text part with the image representations. Right. So the high-level structure of these models is that there's going to be a text encoder. Essentially, in our case, the text encoder is originally pre-trained, and we're going to fine-tune it over these kind of specific pathology texts, like from the tweets itself, to extract information from those tweets. There's also a pre-trained image encoder that we're also going to fine-tune on top of these histology images. And then we need to align the representation that's learned from the text part and from the image part. And to do this, we're going to leverage the tweets because that's where we have the paired information, right? We have the matched text and then the corresponding dialogues, right? To help us to do this alignment between text and image, right? And the idea of this alignment is basically to try to leverage these kind of contrastive learning, right? Where we look at the image embeddings, like the I's here correspond to image embeddings and T's are the text embeddings, right? We want the image embedding and the corresponding text embedding from the same tweet to be closer to each other in this representation space compared to image and text embeddings from different tweets, right? So that's basically the objective of the contrastive learning, to help us to learn like an adapter to align the image and text representation spaces. So that's the methods that we call PLIP, right? And this is basically what this PLIP model would look like. So we are now using it with our colleagues here at Stanford in the pathology department, where our pathology colleagues, if they encounter a challenging image, they will upload that image into the model, right? And then PLIP will first perform some sort of description around what is in that image, right? In this case, it says that this is going to be like an H&E image of the kidney. There's about 700 cells in the image, and there's some inflammation and necrosis, right, in the image. So it first provides this description around morphology, right? And then the clinician can ask questions about the image, right? And the model will take the image and then try to respond and provide some interpretations based on those questions. So in some cases, we found that the pathologists, our clinical colleagues, they may not fully trust the generation of the model itself, right? Because of these risks for hallucinations. So to address that, we actually built a second use mode, more like a retrieval or RAG-based usage mode, where in the retrieval mode, right? So the PLIP model would look at the input image and then actually first do a search across all of the Twitter and other databases. It could be like your internal EHR to retrieve the most similar and the most relevant images that are the most relevant to this input image. It also pulls the corresponding text descriptions, discussions, interpretations of those relevant images, and then summarizes all of that information as a response to the user. And a lot of our clinicians found that to be quite helpful, because even if they don't trust the generation of the model itself, they found that they would still like to see how did their colleagues discuss similar images before, right? So they really like to see this kind of summaries, retrieval and summarization mode. So this is how the clinicians are using PLIP. And it's really can be used as sort of a semantic search engine, right? So for example, some clinicians, they just want to see, okay, they could do some sort of text search. They want to see, okay, particular kinds of neurons, pyramidal neurons in the hippocampus. And then flip model is able to pull up relevant images along with interpretations of those images from other experts. People can also use this to do image-to-image search, right? People can upload an image that they would like to interpret, right? And the model then will pull up relevant images along with their corresponding previous interpretations of these previous images, right? That's also then helping the clinician to interpret new images. Right, so the search engine is now available online, freely available, so that people can use this. So for machine learning researchers, right, so the way that they're using this flip model is really as a sort of a foundation model, right? So the way that's used is that it essentially extracts the representation that's learned by the flip model, right? And then building some additional machine learning classifiers on top of that frozen representation for specific diagnosis or classification tasks. And we showed that across a variety of different diagnosis applications, right? So each column here corresponds to like a different setting, different data sets. By using this frozen representation from Flip, right, and building a simple ML model on top of that, you can actually get higher accuracy compared to some of these really previous state-of-the-art like multitask learning or representation methods that are really trained to do this representation learning specifically for pathology. Right. We also have a bunch of other experiments to show that if you actually start with the flip model as a foundation and do fine-tuning on top of that, you achieve much more efficient, much faster learning using smaller number of samples compared to if you start with like other foundation models that are trained on pathology images or natural images, some of these vision transformers that are just trained on much larger data sets. So just to summarize this part here, right? So this is one of our efforts in building like a chatbot assistant for clinicians, for pathologists. I think the key really insight here is that first we have to realize that there's a lot of to build this kind of chatbot, you really need to have some good demonstration data of how experts interpret these images, right? And it's actually hard to collect those kind of demonstration data, but we found that actually people do a lot of these interpretations on social media, right? And you can get hundreds of thousands of these cases where people interpret and have their friends interpret these images and have discussions. That ends up being a really great data resource to help to align these models, right, to be useful to a pathologist. Okay, so we talked so far about how to use generative AI to help researchers and clinicians. It's also really important to think about how do we build these tools to directly interface with patients, right, to help patients. And I think one of the big problems that we've encountered with patients is that a lot of the medical information ends up being very technical, right, and it's difficult often for patients to understand. Right. So motivated by this, we've been working on how do we make different kinds of medical information easier for patients by using these generative AI and large language models. Just to give you a concrete example of this. Right, so when patients go into hospitals to take part of an operation like a surgery or to participate in a clinical trial, right, they have to go through and sign these consent forms, right? So that's like an integral part of the healthcare process. And here's like its example consent form, right? The details are not so important, but the key thing here is that if you actually look at this consent form, it's not that easy even for us to really read and understand what's going on here. There's a fair amount of like technical, both legal and medical jargons, right? And most of these medical consent forms are pretty difficult to understand, which means that most of us, right, we don't actually try to read these consent forms in a lot of detail. And these forms are really written at college or post-college levels, right? But a good number of Americans actually read more at middle school level, right? So if people don't really read or they don't understand what are in these consent forms, right, that I think really brings up a fundamental question is this medical system really informed consent, right? Informed consent is sort of the basis, right? The underlying foundation across many of medicine, right? But if people can't really understand these forms, if they don't understand these forms, then that sort of calls into the question of this foundation. So what we want to do then is to basically see, can we use large language models to help us to simplify these consent forms to make it much easier for patients to understand, right? And here we basically start with these consent forms, right? The existing ones that have a lot of more jargons, right? And we trained a particular version of GPT-4, right, to simplify these consent forms, right? And with the goal of transforming them, right, rewriting it into a level of about middle school or 8th grade reading level, so that most patients will be able to understand these forms. And here, it's really important for us to do further evaluation, right? So then we had a panel of legal and medical experts, or these who could be medical malpractice lawyers and different clinicians, to actually review the form that are rewritten or generated by the large language model and compare that with the original consent forms to ensure that first, these are factually accurate, and second, that they don't miss any of the key information. So we have this sort of review cycle to validate by experts each of the consent forms produced by the AI model. And the key thing I want to notice here is that the AI model is not generating the consent forms from scratch, right? Their job is to basically just rewrite existing consent forms. And I think that actually also reduces the risk of hallucination because they have a very clear grounding and reference, right? And it's mostly just about simplification rather than generating new content. So here's an example of what a simplification would look like, right? So here's a couple of paragraphs extracted from these original consent forms, right? It just has a fair amount of jargons and it's not very direct or to the point. And after the simplification, here's what the corresponding text will look like, right? It's much shorter and just explains in plain English, right, what is going on here. And we've done a bunch of different experiments to validate this, right? So basically, after you do the simplification with GPT-4, the amount of reading time it requires to read each of these forms is dropped substantially. The reading level, right? So basically, it's the indication of what is the level of education that's required to understand the form, right? So higher, the number here corresponds to more advanced education is required. So lower is better. So after we do the simplification, we dropped from sort of postgraduate level education to about middle school level education. And then the reading ease is basically measuring how easy is it to comprehend the consent forms, right? So after do the simplification, it's also much easier for patients to comprehend. And the part that I'm most excited about is basically as of end of last year, September last year. So our AI-generated consent form are now actually being deployed at large healthcare systems, for example, at Lifespan, which is Rhode Island's largest healthcare system. So every time now the patient goes into surgery, instead of looking at these more complicated consent forms that they had before, now they're actually looking at our language model simplified consent form, right? So this is, I think, one of the earlier examples, right, of actually a deployment of these large language model products in a large healthcare system. So we're quite excited about the impact of this going forward in simplifying other aspects of medicine, including like clinical trial enrollment, patients' FAQs, right? So I think there are a lot of aspects of medicine that this is relatively low-hanging fruit to simplify and to make more accessible. Okay, so with these three examples, right, I hope to give folks a sense of how we try to use this generative AI for researchers, for clinicians, and also to help patients. So maybe I'll just mention one last thing that's a little bit more technical is that, behind each of these tools, we have to make many calls to large language models, and that actually becomes quite expensive, right? It's financially expensive because every time we make a call, right, you have to pay GPT-4 some amount of money, but it also just uses a lot of energy, right? So it's experimentally expensive. And we've been also doing a lot of work thinking about how do we just really substantially reduce the cost of using these large language models. Right, so here's basically like a snapshot of the landscape of this, right? So the x-axis is sort of like essentially the cost of using language models. So not the training cost, right, but it's actually the inference cost of using these models. And the y-axis is basically like a metric of some level of performance against different benchmarks. You can see here that GPT-4 is the most performant model, but it's also the most expensive one to use, right? On the other hand, here you have some of these cheaper models, but their performance is also more variable. So we've developed a system that we call Frugal GPT, which is a way we think of trying to optimally trade off the cost and performance of these different language models, right? So Frugal GPT is able to actually achieve even a slightly better or comparable performance to GPT-4, but it really uses only about 2% of the cost and 2% of the energy consumption as GPT-4. And the key idea here is that Frugal GPT is actually not a single language model, but we view this as basically like a compound system over this whole ecosystem of language models, right? So the idea here is that Frugal GPT will look at different questions, right, that the user would give. And some of the simpler questions, right, if the question is relatively simple, you don't actually need to use the biggest language model to answer that question, right? You can triage those questions to some of the smaller models and get a perfectly fine answer. So you want to save the most difficult and challenging generations, right, for the large language models, for the biggest models. So if you can actually do that kind of triaging efficiently, that can actually lead to a lot of the cost savings. So that's the main idea of Frugal GPT. It's a very small language model that we trained to basically learn what we call like this language model cascade, right? So for a given query, the Frugal GPT will construct a cascade of language models from small to large, based on what it thinks that query's difficulty is and areas of expertise of these different models. So first, maybe it will send this query to a very small model, and it looks at the output of the model as well as the confidence of that model and see, is this good enough or not? If it's not very confident or not very satisfactory, then it passes that on to a medium-sized model and finally onto GPT-4. But in many of these instances, for many questions that patients or other people might have, the smaller models are actually reasonable at answering these questions, right? So we save a lot of costs because we don't actually go all the way up the chain, the cascade to GPT-4. And the nice thing here is that this is all done adaptively, right? For different queries, depends on different topic, there could be different cascades, right? The model, Frugal GPT, has learned on how to optimize the trade-off. So this is we applied it to a bunch of different settings to show that, in different settings, you get different cascades and different trade-offs. But in all the cases, it's actually able to save quite a lot of cost compared to always using GPT-4 or always using the single best language model, right? So across different settings, it's able to achieve like a cost saving about 50 to 98% while matching the performance of the single best language model, which is often GPT-4. In some cases, like we see here, it's able to actually do better than GPT-4 because it learns that for certain kinds of questions, some combination of these smaller language models or more specialized models are actually better at answering than just GPT-4, right? So that's why it's able to even do better than the performance of always using the single language model. So this is sort of the overall direction that I'm quite excited about on the more ML side, right? And with a bunch of great colleagues, especially Matej Zaharia, we had a blog post on this earlier last week, where I think really where the field is going now is shifting from individual language models, individual foundation models, but to what we're calling these compound AI systems. So Frugal GPT is one example of a compound AI system, right? I view this as really like a middle layer, like sort of like Expedia, right, for language models. Instead of looking for, when you try to buy flights, you don't go to individual airlines anymore, right? You go to like this middle layer, like Expedia or Google Flights, which optimizes your constraints across all of these different airlines. Right? So that's what we're trying to build here, right? Which is Frugal GPT that optimizes your budget constraints and your requirements across all of the existing language models. I think that's sort of where the industry and the field seem to be going forward. Okay, so I'll just wrap up here. So what I really want to highlight here is to give people some examples concretely of how we use generative AI to benefit researchers, clinicians, and patients, right? So we talked about how we can generate new molecules really as a way to expand the search space for drug developers, and how we can use these vision language models, right, to build assistance to pathologists, to clinicians. And from the patient side, how we try to use these generative AI tools to really simplify medical information for patients, right? And this really worked a lot by fantastic collaborators, and especially by students I'm fortunate to work with here at Stanford. So I'm happy to stop here and take any more questions.
Thank you so much, James, for an amazing talk. what a tour de force.
Thank you. I would like to ask about the ethical side using AI for pathologists. Do you have any comments?
Yeah, I think that's an interesting question. So I think for pathologists, we talk to a lot of them, to a lot of our collaborators, because the key thing that they want to know about is that essentially how reliable is the algorithm across different settings, across different types of patients, right? Because often patients may be from different demographic groups or from different backgrounds, right? Will the algorithm be able to provide reasonable responses across these diverse settings? Right? That's one consideration that they're very interested in. And the second thing is that, would the algorithm leak private information that are very sensitive to a particular patient? All right, so I think those are really both important considerations. The way that we try to address them in our deployment here at Stanford is that for the first one, this is actually motivation for why we end up doing this retrieval model, right? So oftentimes the doctors, they like to see how did their colleagues have discussed these different kinds of patients before that are relevant, right? They don't just want to see the generation of the AI chatbot itself, but they want to see the human colleagues' discussions, right? So that's why we do this retrieval part to try to summarize and bring in all of these other sources of information for the doctors. And then the way that we try to handle the privacy aspect of this is that this is all done like locally, right? So we actually made this PLIT model open source, so anyone can actually run that locally. So that the model doesn't have to leak out or release any information online.
Hi, yes, I'm here.
Fantastic. Would you mind asking your question?
Yeah, sure. So I was working with a lot of like electron microscope images, which are very fluctuating. So my question is just how accurate the ground truth images from Twitter are. How do you collect it? And after collecting, do you do a lot of manually inspection, and are there any steps you do to like optimize the ground truth picking features of the images for the model?
Yeah, thank you for that question. That's a good question. So first thing to note is that the images posted on Twitter tends to be the harder images, right? Because if it's like relatively easy image, then people don't need to ask their colleagues for help to interpret, right? So they don't actually post those online. So it's almost like basically the clinicians have done their own active learning to try to find the most ambiguous and hardest, most challenging images. And then those are the ones that are posted online, right? I think from that perspective, actually, these are hard images, but they are also quite informative for training the AI model. And the annotations, discussions of those images can be very noisy. Right? So the way that we try to filter these is to basically, we first generate our own expert labels, right? And then we train, because there are millions of these images, then we have to train our own machine learning algorithms based on our expert labels of the quality of each of these image. Right, so that's how we build like an image filter. And then we also have experts to label the quality of the text of the tweets itself, the discussions, and then we train like another text classifier. So we do a few rounds of those filterings. We also use things like the number of likes, right? A comment gets as some additional proxy signals. So we do all of these filterings to end up with, I think, a higher quality set from the initial set of tweets.
Great, thank you.
Thanks.
James, I'm having a question myself, if you allow me. And the question is actually about the last set of slides. So you were talking about your recent blog. Would you mind saying, unfortunately, I didn't read it yet, so would you mind saying a few words about it? Frugal GPT sounds, I did read about Frugal GPT and I was very intrigued, so I'm keen to hear about your thoughts in the recent blog. Would you mind saying a few words about it?
Yeah, absolutely. Yeah. So I think the main idea we're discussing in this recent blog, I think is quite intuitive and quite simple. Is that we're seeing, especially in industry, a really shift where instead of relying on a single language model for a particular task, now more and more companies have to rely on like a system of language models, right? And maybe they use like Llama to generate some initial responses, that they have GPT-4 to check some of those responses, and then they maybe have like another layer, another language model to do some additional cleaning. So it's actually getting into quite an interesting like a network of language models that are also checking, validating, or providing feedbacks to each other. And when we say the compound AI system, we're referring to that whole network of interacting language models, right? That are, some of them are like evaluation, others are doing more generation, and there's different feedback loops. And I think that kind of compound AI system is really interesting because there are even just a lot of fundamental open questions around how do we design these kind of compound AI systems. And I think there are a lot of low-hanging fruits in that, let's say, even if we don't improve the individual language models anymore, right? But I think by coming up with better designs of how we put these networks together into different cascades or different pipelines, you can still actually get substantial improvements in reliability and performance at a much reduced energy consumption and financial cost. Right? So I think that's sort of the research agenda that we want to promote people to look into, because I think there's actually a lot of opportunities of that, especially in large-scale deployments.
Definitely. Fantastic idea. And ensembles in general, we know from many years that they are better than individual models. So I think even more a chance to do something interesting here. Okay, so we have, maybe it's 5 o'clock, so I think we have time maybe for a very, very final question, if your time allows, James. So I see that Andy put his hand on. So Andy, maybe a final brief question from you.
Yeah, thanks, James. Just maybe a stupid question, but in the first part of your talk, you described how after discovering penicillin, there was a kind of a gold rush for 20 years, where a bunch of new antibodies were discovered. And so obviously what you presented here might allow us to find many more very quickly. But do you view that number of potential molecules as effectively infinite that would be useful, or will we just run out again, like in some fairly short amount of time?
Yes, it's an interesting question. So we've calculated sort of the accessible space of potential new molecules, right? And our estimate is that there's actually potentially several hundred billions of new molecules that we can relatively easily synthesize, right? Using this kind of recipes generated by the AI model that haven't actually been seen before in nature. And among that space of hundreds of billions of molecules, I think there's actually a lot of really interesting drug candidates for a lot of different diseases. I think we're not close to saturating the space of new molecules.
Thank you very much, James, for your fantastic talk and many answers. And we will be putting this video online, and I'm sure many more people will get engaged with your agenda, also locally here in Cambridge. So thank you very much for making the time to give this fantastic talk and inspire us.
Thank you very much. Great to see everyone.